Hi, hello, good morning. This is T. Ramudu. I am studying 9th class and I am from TGTWRJC Boys, Huzurnagar. Generally, all the living organisms on the earth is made up of cells. For example, plants, animals, humans, etc. And today we are going to discuss about plant cell and its ultrastructure. It is living and eukaryotic cell. In this, the first one is cell wall. This is a cell wall. It is flexible and it is made up of cellulose. It gives definite shape and gives protection to the cell. It is the outermost layer of the cell. It is permeable to all kinds of materials into the cell and vice versa. It exerts inward wall pressure to resist outward pressure exerted by the cytoplasm. It is unique feature seen in plant cell. It is considered to be one of the major differences between plant cell and animal cell. I am going to discuss with you the cell membrane. This is the cell membrane. It is flexible and it is made up of lipids and proteins. It defines shape and size to the cell. It encloses the cytoplasm and protects it from external environment. It is outermost layer in animal cell but not in plant cell. The cell membrane plays a crucial role in maintaining the balance of substances, the exchange of substances. So the cell membrane is very selectively, hence it is called as selectively permeable membrane.
Good morning, this is B. Suresh, 9th standard from TGTWRJC Boys, Huzurnagar. Now I am discussing with you the cytoplasm. This is cytoplasm. Cytoplasm is the fluid content bounded by the plasma membrane. It contains many specialized cell organelles. Cytoplasm is main site for biological activities. Now I am discussing the second cell organelle is chloroplast. This is chloroplast. These are green colored plastids. They mainly contain green substance called chlorophyll. It doubled membrane cell organelle. It consists of telocares into a pack called granum. It also having 70s ribosomes and a small amount of DNA. It also called kitchen house of the cell. Thank you.
Hi, hello, good morning. This is V. Sandeep, 9th class, TGTWRJC Boys, Huzurnagar. I am discussing with you about endoplasmic reticulum. This is endoplasmic reticulum, a network of membrane near nucleus in cytoplasm is called endoplasmic reticulum. It is similar to plasma membrane structure. These are first discovered by K.R. Porter. The endoplasmic reticulum have some granular structures on its surface called rough endoplasmic reticulum. This is rough endoplasmic reticulum. The rough endoplasmic reticulum is the site of protein synthesis. The endoplasmic reticulum do not have granules on its surface called smooth endoplasmic reticulum. This is smooth endoplasmic reticulum. The smooth endoplasmic reticulum is the site of lipid synthesis. The granules are ribosomes. This is ribosome. Attached to endoplasmic reticulum. The function of endoplasmic reticulum to serve as channels of transportation of materials between nucleus and cytoplasm. In liver cell, smooth endoplasmic reticulum is the help to detoxification of poisonous substance and drugs. Continue with Golgi operator. These are Golgi operators. Camillo Golgi first discovered this reticular structure near nucleus. It consists many flat disc-shaped sacs called cisternae. These are stacked parallel to each other. It performs the function of packaging materials to be delivered either intercellular targets or secreted outside of the cell. It also makes glycoproteins and glycolipids. In plants, it involved in synthesis of cell wall materials. It also plays a role in formation of cell plates during the cell division. Thank you.
Hello, good morning. This is N. Sai Kumar from 9th Standard and from TGTWRJC Boys, Huzurnagar. Now I am going to discuss with you is the mitochondria. And this is the mitochondria. These are small spherical or cylindrical cell organelles. These are 2 to 8 micron long and 0.5 micron wide. It is about 150 times smaller than the nucleus. There are about 100 to 150 in each cell. It was first discovered by Altman and named as bioplast. Later, Benda named as mitochondria. It is double membraned cell organelle. Inner membrane holds inside called cristae. The space between cristae filled with substance called matrix. Cristae having oxysomes or elementary particles involved in electron transport system. Mitochondria is responsible for cellular respiration. Matrix of mitochondria having enzymes for cellular respiration. Through cellular respiration, cells get energy to do work. Hence, the mitochondria also known as the powerhouse of the cell. And the mitochondria also having the 70 steps of ribosomes and the small amount of DNA. I am going to discuss with you another cell organelle is the lysosome, and this is the lysosome. One of the facts that troubled the scientists for a long time was that certain enzymes present in the cell have the ability to destroy almost all the structures in the cell. This puzzle was solved when lysosomes were discovered as tiny particles visible in the cytoplasm. It was found that they contained the destructive enzymes. These enzymes normally do not come in contact with the rest of the cell. The material not need to be contained, destroyed or transported to the lysosome.
Hello, good morning. This is Bhimanishwa and I am standing from TGTWRJC Boys, Huzurnagar. Now I am discussing with you is the ribosomes. This is a ribosome. This is small granule-like structure, first discovered by George Faraday. They are formed of RNA and protein. There are two types, 70S ribosomes, 80S ribosomes. Some are freely in cytoplasm and some are attached of endoplasmic reticulum. They are sites for protein synthesis. 70S ribosomes also found in chloroplast and mitochondria.
Hey, hello, good morning to one and all. This is Siddhartha from TGTWRJC Boys Huzurnagar School. We are discussing about microbodies. This is peroxisomes. This is glycosomes. Peroxisomes and glycosomes often called microbodies. These are membrane-bound and spherical-shaped minute cell organelles. Peroxisomes are involved in catabolism of long-chain fatty acid and play important role in synthesis of phospholipids. Peroxisomes are also involved in photorespiration and protection of cell from toxic effect of hydrogen peroxide. Glycosomes are generally found in fat-rich germinating seeds. Glycosomes are contain enzymes of glycolate cycle which convert stored lipids to carbohydrate.
Hello, this is C.H. Ganesh. I am studying 9th standard, TGTWRJC Boys Huzurnagar. I am telling you about now cytoskeleton. This is cytoskeleton. An elaborated network filamentous proteinaceous structure in cytoplasm of cell is called cytoskeleton. It contains three major components. First, microfilament and intermediate filament and microtubules. Cytoskeleton involved in many functions such as mechanical support and maintenance of cell shape and cell motility and intercellular transportation and signaling across the cell and karyokinetics. Thank you.
Hi, hello. Good morning to one and all. I am Dita from 9th standard in ECW URJC Boys School, Huzurnagar. Now I am going to discuss the topic is vacuole. Vacuole. The large empty space present in the cell or vacuole. This is full-filled sac-like structure in animal cell which are small and large in plant cell. Vacuole maintains Serge pressure within the cell. They export unwanted substance from the cell.
Going to discuss with you the nucleus. This is the nucleus. Dark colored spherical shaped structure near the center of the cell is called nucleus. It was named by Robert Brown in the year of 1831. This is one of the most important cell organelles in the cell. Nucleus is also called as control room of the cell. It is obtained in prokaryotic cell like bacteria, orbits of mammals, prokaryotic tube cells in plants. Nucleus regulates and controls all the function of the cell and determines the characteristics of an organism. Nucleus have genetic material called DNA. During the cell division, chromatin material condensed and formed into rod-shaped structure called chromosomes. Chromosomes carry the genetic information from their parents to their offspring. This is the spherical shaped structure is called nucleolus and this is called nucleoplasm.
Project done by all my friends as a team. I call my friends, please come. For this project, our botany lecturer, mentor guide and advisors and director Sri P. Ramakrishna Sir, JL in botany, helped us. Now I call up them. Sir, please come here.
Thank you. So my dear students, thank you. So right, we done this project work with this help of the student and they helped in many ways. So the project was very successful and next upcoming days we make this kind of biological models to learn the students very easy. So we do best. Thank you. Done.